I'm going to talk about a phenomenon called cross-frequency coupling and the role of non-sinusoidal oscillations in identifying cross-frequency coupling. And then I'll argue that a multivariate approach is much better for various reasons. So what is cross-frequency coupling? Well, cross-frequency coupling refers to a relationship, a statistical relationship between activity in two different frequency bands. Here you see an example of cross-frequency coupling. So there is this slower rhythm here, and then there's something happening with this faster rhythm such that the faster or the amplitude of the faster rhythm is larger when the slower rhythm is at its peak, and then this faster rhythms decays during the troughs of the slower oscillation. So this is a manifestation. This is an example of cross-frequency coupling because the phase at one frequency, at the slower frequency, is modulating the amplitude or the power at this faster frequency. I'm going to talk a little bit more later on about the biological theory, the neurobiological theory of cross-frequency coupling, but the general idea is that in the brain, it could be that there are rhythms that are kind of regulating or clocking some computation or setting up some environment for computation, and the actual computations are being performed by these faster rhythms in smaller, possibly more local networks. So as I mentioned, this is just one characterization, one way of expressing cross-frequency coupling. This is called phase-amplitude coupling because the phase of the slower rhythm is determining the amplitude of the faster rhythm. This slide shows that, in fact, there are many different ways to conceptualize the manifestations of cross-frequency coupling. And it's basically all possible combinations of power in one channel and phase in another channel or frequency. So power, phase, and frequency, and you can change these things around and get different manifestations of cross-frequency coupling. Now, that's not to say that all of these forms of cross-frequency coupling happen in the brain, and this is not to say that these are physiologically meaningful and interpretable methods of implementing computations in the brain. But this is just the realm of mathematical possibilities for the way that two different signals can interact across different frequencies. Now, there's a bit of a problem with most of the cross-frequency coupling methods that have been proposed in the literature. And that problem is that cross-frequency coupling methods do not necessarily measure or do not selectively measure cross-frequency coupling. In fact, most of the measures of univariate cross-frequency coupling, and univariate cross-frequency coupling just means that you're looking at cross-frequency coupling within the same channel, within the same signal. They actually cannot really distinguish between real cross-frequency coupling, like what we would think of as physiological, biological cross-frequency coupling, and harmonics or dynamics resulting from non-stationary narrowband activity. I'll provide a brief definition of non-stationary narrowband activity in the next slide. So this is difficult. This is a tricky situation because it makes it hard to distinguish between true cross-frequency coupling and non-sinusoidal rhythms without any further data investigations. I'm going to explain why this is the case in a moment. So let me first start by just introducing this concept of stationary versus non-stationary. So for a narrowband signal, stationarity just means that the amplitude and the frequency of the signal is not changing over time. Basically, when you think of stationarity, you can think of a pure sine wave as being a stationary signal. So here's what it looks in the time domain, and here's how it looks in the frequency domain. It's just a spike at the frequency. A non-stationary signal has some variance, it has some variability, so it's changing over time. The amplitude is changing over time, the frequency is changing a bit over time, maybe there's some noise in there, and that has a power spectrum that looks a little bit wider. So it still has a peak at the right frequency, but you can see it's more spread out. It covers a lot more, a much larger range of frequencies compared to this stationary sinusoidal rhythm. Now, if you've worked with biological signals and neural signals, then you probably will immediately recognize that this is much more physiologically plausible. This is more biologically realistic. Of course, this is, not an actual biological signal. I just generated this. But this looks more plausible, and this, I wouldn't argue that there are no pure sine waves in biology, but certainly biology is characterized by variability and by dynamics and change. Okay, so it's clear to people who've worked with biological signals that this is more reflective of the reality of what we see in neural time series data. So what you're looking at here is a comodulogram. It shows frequency for the phase and frequency for amplitude. And the color here corresponds to an increase in the relationship between the amplitude or the power at this frequency and the phase of this lower frequency. Now, if there is no cross-frequency coupling in the signal, zero cross-frequency coupling, then this entire plot would be blue. So the fact that we see some yellow-orange stuff here tells us that there's some cross-frequency coupling that we have identified in the data between 6 Hertz and 40 Hertz. Okay, so that's just to briefly explain what this plot means and how to interpret this plot. So here you see 3 different comodulograms for 3 different signals, and here you see 3 signals. Now I've done something tricky here. I've done something sneaky. I've actually mixed up the order. So it's not the case that this signal has this cross-frequency coupling comodulogram. So it could be any one of these, and this could be any one of these, and this could be any one of these. So because you're watching this online, this is a great opportunity. You can pause the video and see if you can guess which signal corresponds to which comodulogram. So the answer is that it doesn't matter. All these different signals have pretty much the same comodulograms. They look a little bit different, but I could have tweaked the parameters here when generating these signals to make these 3 plots look more similar to each other. And that's particularly striking for this case where, when you look at this sine wave, you wouldn't say that there's cross-frequency coupling happening in this signal. It just looks like, the amplifier clipped or something. There's something a little weird happening at the tops of these otherwise pure sine waves. And yet it produces a plot that looks like this, which if you didn't see the time series and you just saw this plot, you would say, hey, this is beautiful cross-frequency coupling. So this is an illustration of what I meant. Now, I created these signals, and I created this one to be kind of, it's difficult to say what is real cross-frequency coupling, particularly in simulated data. But the way that I generated this data time series is by taking 2 distinct signals. So I simulated one signal in theta and one signal in gamma. And then I was kind of simulating that these 2 signals are interacting with each other. And then I projected them both to an imaginary electrode, a virtual electrode. So in fact, this signal is a mixture of 2 different local field potentials. That's the simulation assumption behind this signal. And this one you can see is just a pure sine wave that's clipped. And here, this is the same signal that I showed in the first slide. And this is, also, this isn't simulated in a way that reflects what we think is happening in the brain when the brain exhibits cross-frequency coupling. All I did was take one sine wave and use it to define the amplitude of another sine wave. Okay, so let's assume for the moment that this is cross-frequency coupling, and these 2 reflect not cross-frequency coupling, but just dynamics in a signal. So then you can see what the problem is. We cannot just look at the cross-frequency coupling result and determine which is reflecting true cross-frequency coupling and which reflects non-stationary features of the signals. Okay, so just to reiterate, the point here is that univariate cross-frequency coupling analyses are actually measuring non-sinusoidal waveform shapes. They're not actually measuring cross-frequency coupling exclusively. If there is cross-frequency coupling, these cross-frequency coupling measures will detect it. So there is a positive case there. But if there's no true cross-frequency coupling, but there are non-sinusoidal waveform shapes, then most univariate cross-frequency coupling measures will also give a positive result. So that means that results of a univariate cross-frequency coupling analysis method can be difficult to interpret. So what's really happening in the literature when we do this as scientists, as researchers, is we apply these univariate cross-frequency coupling analyses, and then we infer that a non-sinusoidal waveform indicates cross-frequency coupling. So this is an inference that we make when we see our univariate cross-frequency coupling results. So we say positive univariate cross-frequency coupling equals neural cross-frequency coupling. Now, that could be the explanation, but it's also possible that a positive univariate cross-frequency coupling result is driven by a non-sinusoidal waveform shape, not by true cross-frequency coupling. Okay, so the problem is that I'm going to argue now in the next slide that this assumption is probably not such a great assumption. I think what's really happening is that when we get our univariate cross-frequency coupling results, they reflect a mixture of true cross-frequency coupling and non-sinusoidal waveform shapes. So I wouldn't argue, certainly don't want to argue that univariate cross-frequency coupling methods are wrong. The point is that they are not as selective to cross-frequency coupling as we would really like them to be. Okay, so why do I claim that this assumption is probably invalid, or at least a little bit too far? It's a little bit inappropriate. Well, that's because we know from a lot of research collected over many decades that brain rhythms, or electrical brain activity, is rhythmic. It exhibits strong rhythms, narrowband frequency activity, but not necessarily pure sine waves. Here you see two examples of this. These are brain rhythms that are definitely non-sinusoidal, but they are clearly rhythmic. Now, these are not, some of these are even simulated data. These are not necessarily the best examples or the only examples. These are just the first two pictures that came up when I was searching for this on Google. So I decided to use these two pictures. And then I spent about 3 minutes looking on PubMed, and I just picked out the references that also show that empirical brain activity, so real brain activity, is rhythmic but non-sinusoidal. Again, this is not an exhaustive list. There are many more papers than this. These are just, the 5 papers these are the references that I recognized immediately because I'm familiar with these papers after like 3 minutes of searching. So there's a whole literature that goes back decades showing that electrical brain activity is rhythmic but non-sinusoidal. So again, I want to make sure I'm being very clear about this because I don't want to say that univariate cross-frequency coupling measures are invalid per se and shouldn't be used. The problem is that they are not exclusive to measuring cross-frequency coupling. I think that what they actually measure in reality is the extent to which a rhythm deviates from a pure sine wave, and I suspect that all of the findings in the literature based on univariate cross-frequency coupling measures, including my own, by the way, I'm happy to criticize my own previous work, those reflect some combination, a mixture of non-sinusoidal rhythms and actual, real, genuine cross-frequency coupling. So what can we do about this? How can we fix this problem and come up with a better measure of cross-frequency coupling that also is robust to the non-sinusoidalities that we know are going to be in this signal? Well, I think a good way to approach this is by not thinking about the methods first, but thinking about the biology, thinking about the theory. So again, the idea of cross-frequency coupling in the brain is that there is some network that's oscillating at some slower frequency, let's say theta, and that network oscillating at a slower frequency is regulating the activity at faster frequencies, and those should be in different networks. So that's my key assumption that I'm starting from. So my key assumption here is that the low frequency networks are not completely overlapping with the high frequency networks. So whatever brain networks, circuits, brain regions are generating this slow rhythm, those are not exactly the same neurons. They can be overlapping, but they are not exactly the same neurons as the neurons that are generating this faster rhythm. So that means that these two networks, I'm thinking about these as two networks, the low frequency network and the high frequency network. and they're at least partially distinct. So they can recruit overlapping neurons, but at least some of the neurons are different from each other. So the implication of that is that the electrical fields generated by these two networks are going to be separable. They're going to be linearly separable. They can be spatially overlapping. You'll see examples of that in a few minutes, but I'm assuming that they are going to be not identical. Now, if you're only measuring from one electrode, you cannot distinguish between those different networks because one electrode is measuring a mixture of the signals, the local field potentials from these two networks. But if you have many, many electrodes placed, spatially placed around these networks, then it's going to be possible to linearly separate the local field potentials generated by the slow network from the field potentials generated by this faster network. Okay, so that's going to motivate, so it's starting from the theory, basically working from the theory to the analysis, motivates the use of a multivariate analysis method that allows for linear separation of spatially overlapping networks. Okay, so that's the basic idea, and now I'm going to jump into a set of methods that I worked through a couple of years ago. But before telling you about that particular method, that my particular way of solving this problem, I just want to mention that I think this assumption is really important, and this approach of starting from the theory and working from there to the analyses, as opposed to starting from the math or starting from signal processing and then trying to develop an analysis method. I think that's the good approach. I think a multivariate approach is what we need for cross-frequency coupling, and I don't want to argue that the particular method that I'm going to show you is necessarily the best or necessarily the only one. Okay, so with that said, what we need to do is come up with a compact representation of all the interactions between all the channels in our data set. And we do that using a covariance matrix. So here we see some simulated data. This is, I guess, 20 channels over time. And you can see that there is some, there's some spatial dynamics. There's some correlations across the different channels. So we can compute from this data matrix a covariance matrix, which has channels by channels on both axes. And basically each block in this matrix, each little pixel, corresponds to the linear relationship between this channel and this channel. So covariance matrix is really great because it encodes all of the pairwise interactions or the linear interactions between all of the different channels. Here you see two covariance matrices from real data. So the previous slide was simulated data. So these are from real data, and it's from around 224, I think it was, channels. And these covariance matrices are, first of all, just really beautiful to look at. But when the channels are numbered in some kind of meaningful sense, you can also immediately see interesting patterns emerging in the covariance matrices. In this case, these were electrodes implanted in 4 different brain regions. So you see strong coherence within each brain region and some cross-coherence across the different, or covariance, I should say, across different sets of regions. Now, this is another covariance matrix that comes from the same data set. So it's the same channels, the same animal, same data set, but this is just taken from a different feature of the data. So that can be a different time window, for example, or a different frequency band. So these come from the same data. You can see that the large scale features of both of these plots are the same, but they differ in quite a lot of detail. That's pretty easy to see in some cases. Here's another striking case where you see some covariance in this feature of the data that seems to be absent in this feature of the data, and so on. So I'm going to call this covariance matrix R. This is like a reference covariance matrix. And this I'll call S. That's like a signal covariance matrix. So the idea of doing this covariance-based separation method is that we want to find some pattern in the data that distinguishes this covariance matrix from this covariance matrix. So how is this matrix different from this matrix? Now you might think of just subtracting these two matrices from each other. That's going to give you another covariance matrix that is the difference between these two. But that's not the right thing. That's not what we want to achieve. What we actually want is a set of weights for each channel, so a number that we can assign for each channel, such that when we combine all the channels together according to those weights, that is a feature or a pattern in the data that best separates this matrix from this matrix. So this is the general idea. We want to find a vector, a weighting of the channels that best separates this matrix from this matrix. So now I'm going to walk you through a little bit of the math, although I'm skipping a lot of the interim details. So we want something that's kind of like this. We want to compute the ratio between the S covariance matrix and the R covariance matrix. Now, division is not really defined for matrices. So what we actually want is to find some vector. So this is vector W, where the length of this vector, the number of elements in this vector, corresponds to the number of channels in the data set. So we want to find some way of weighting this covariance matrix such that when we apply that set of weights, that is going to maximize this ratio. It's going to maximally separate the S covariance matrix from the R covariance matrix. So you might already recognize this as an optimization problem. So we want to find the vector W that maximizes this relationship. So it turns out that linear algebra gives us a really elegant solution to this maximization problem, which is this formula, which is called a generalized eigenvalue equation, or more generally, a generalized eigendecomposition on these two matrices S and R. So in the interest of time, I'm skipping a few steps. That there's about 5 minutes of algebra that gets you from this to this, which I'm skipping over here. But the point is that we just apply a generalized eigendecomposition on these two covariance matrices, S and R, and what we get back is two other matrices, W and lambda. W is the eigenvectors and lambda is the eigenvalues. And the idea is that the column of W, so the eigenvector that is associated with the largest eigenvalue, is the W here that maximizes this relationship. And that's what we're looking for here. That's going to be our multivariate patterns in the data that best separate matrix S, or the data that went into creating matrix S, from the data that went into creating matrix R. So let me show you what this looks like in pictures. So here we have the matrices S and R. Just move R over to the left side from the right side. And here's the matrix of eigenvectors, and here we have the diagonal matrix of eigenvalues. So the idea is that we start from exactly the same data, and we just take different features of the data, maybe different time windows or different frequency bands, but it's all coming from the same data with the same channels and so on. So part of the data that's treated as a reference period or a baseline period, that goes into matrix R. Part of the data that we're interested in goes into matrix S. And then the end result after the generalized eigendecomposition is to take the eigenvector associated with the largest eigenvalue and use that as a set of channel weights. So remember, we have the number of elements in this vector corresponding to the number of channels, and then we multiply each of the data time series from each channel by the corresponding element in the eigenvector. And then we add all of these up. So in the end, that gives us one single time series. That's like a component time series. This is kind of equivalent to, what you think about with PCA or ICA, in the sense that you get a single component that reflects a weighted combination of all the channels. And then what really differs between generalized eigendecomposition and ICA and PCA, non-negative matrix factorization, and all these other decomposition techniques is the exact formula that you're using, the optimization criteria that you're using to create these weight vectors or these spatial filters. Okay, so that's a bit of the idea, a little bit of the math. And now I just want to show you a little bit of code. So this is MATLAB code, and it basically, computing the generalized eigendecomposition basically involves 3 steps. So we compute the covariance matrices, that's the data matrix times its transpose. Then we implement the eigendecomposition, which is very simple in MATLAB. Of course, you could also implement this in Python. It would be almost just as simple. And then you create the component time series, which is one of the eigenvectors times the data. Now, what's also interesting about this approach is you don't need to multiply the weight by the filtered data, the selected data that you're using to create the covariance matrix. You can actually apply that eigenvector back to the raw data, not the filtered data. So this gives you more opportunities for careful analyses of these component time series. Now, as you might imagine, in doing this in practice is more than 5 lines of code. There's a lot of stuff you have to do, appropriate filtering and selecting to get to these data matrices, and you have to do more analyses and so on. But, I just want to highlight the fact that this is not a terribly difficult procedure to implement. Okay, so this approach, generalized eigendecomposition for cross-frequency coupling, relies on 3 key assumptions. One assumption I've already mentioned, and that is that cross-frequency coupling reflects the regulation of 2 distinct populations of neurons. They can be overlapping, but they cannot be exactly the same. They have to be at least partially spatially separable. They can be correlated, they can be overlapping, but they need to be linearly separable. And if this assumption doesn't hold, then, if it's exactly the same neurons that are generating the low frequency and the high frequency rhythms, then you have to use a different approach, basically. This approach won't be appropriate. And those networks need to be spatially stationary. That basically just means that the networks need to be the same. So it's the same neurons with the same orientations. That's because if the networks are constantly moving all over the brain, if you have like a moving dipole, then the covariance matrices are going to be changing over time. And if the covariance matrices are changing over time, then it's unclear what the decomposition is going to reflect. So the covariance matrices must be pretty stationary. And you also have to have enough electrodes to be able to linearly separate these 2 probably spatially overlapping networks. Now, how many is many? You certainly need more than 1, but how many you need exactly depends on the kind of data you're acquiring and whether it's from outside the brain like EEG or MEG or inside the brain like LFP. So the benefits of this approach is you get a boosted SNR because we're averaging over all the electrodes, but we're averaging over in a smart way. You'll see an example of this in a few moments. It's totally robust to the confounds that plague these univariate cross-frequency coupling measures, as I discussed before. I will explain why that is on the next slide. And there are, or in 2 slides, there are many ways to customize this analysis for specific hypotheses, and I'm going to show you several examples of how to do that. There's a couple of limitations because this is a kind of advanced decomposition technique. It requires clean data, so you might need to do some additional cleaning or preparation compared to a normal set of data analyses. And, this I listed as a benefit on the previous slide, but this general approach of doing generalized eigendecomposition for cross-frequency coupling, it has many options. There's many ways of setting up the analysis, of selecting the data, of filtering the data, of fine-tuning the parameters for your data selection and filtering. So I do not recommend taking this kind of approach as a real exploratory data mining approach when you just want to see if there's something in the data without having a clear idea of what that something is going to be. And that's just because the space of total possibilities is a bit too large. So I recommend approaching your data for a multivariate cross-frequency coupling analysis only when you have a really clear hypothesis that is going to guide you to select the analysis pipeline in an appropriate way. Okay, so this is the same picture as I showed several slides ago, just to illustrate one way of setting up this analysis. So let's say we have our channel data here, and we have this theta rhythm that we've extracted from the data. Maybe this is from one channel, or maybe this is from a component that you've extracted the theta activity from. So now we can take, for the R matrix, the reference matrix, all the data. Now all the data is actually pooling over all of the phases of the theta phase. And or the theta rhythm. And for the S matrix, we take only brief snippets of data around the theta troughs. So what we're doing in this generalized eigendecomposition here is finding a set of weights that maximally differentiates the signal, the data, during the theta troughs compared to the rest of the signal. Now, if you want, you could also select the R matrix data just to come from the periods in between the troughs. That turns out to be more or less equivalent. So the idea here is that we are sub-selecting data from particular theta phases for the S matrix and from all theta phases for the R matrix. So this is going to identify a pattern, a spatial pattern in the data that is maximally differentiating theta troughs from the rest of the theta cycle. So that's one illustration of how to use this method. What I'm going to do now is show you a couple of slides of simulations. Data simulations are great because we can compare the results of the analysis against the ground truth because we know what the ground truth is. And then after I show you some simulations, I'll switch to some proof of principle demonstrations in real data, in empirical data. So what I did here is simulate some data in a couple of dipoles that were all pretty close to each other and projected those dipoles out to the EEG topography. So this is the topography of a component that oscillated in theta. And here are two other components that oscillated in gamma at two different frequencies. I think it was 40 hertz and 50 hertz, but you'll see in the slide in a moment. So you can see that these dipoles have overlapping projections. So that means that at the level of electrodes, you're going to measure from all three of these dipoles simultaneously. And then the idea was that the amplitude of the gamma 1 dipole was synchronized to the theta troughs. And the gamma 2 dipole was just kind of going on its own. It was exhibiting gamma oscillations, but totally independent of theta. So I simulated cross-frequency coupling between theta and gamma 1 and no cross-frequency coupling between theta and gamma 2. So this is the simulated data. Here's the recovered data that I got from the generalized eigendecomposition. So this is for finding a theta peak or theta spatial filter, and you can see that matches the simulation really well. And here I looked for a spatial filter that maximally separates the data from the theta troughs versus the rest of the theta time series, like what I showed in the previous slide. And you can see that that looks a lot more like the gamma 1 dipole and a lot less like the gamma 2 dipole. So here we see some analyses of the component time series that we get from these different spatial filters. So here you see the power spectrum of the two components, the theta component and the theta trough component, which shows a strong peak at gamma. And you see also the side lobes here, because that is how I simulated the synchronization. So these side lobes, the distance of these side lobes is theta. This plot here shows the power spectrum from electrode POZ. So this is from one specific electrode, one channel that's located somewhere around here. So that's going to pick up the signal from all of these three dipoles. They all get mixed together, and a lot of 1/f noise, which I added just to make the signal look a little bit more realistic. So here you see the two gamma peaks. This is the one that shows cross-frequency coupling, which you see here. And this one does not show cross-frequency coupling, and you don't see any of that evidence here. And you can see that these spatial filters also really attenuated the noise. And that's because the noise has a very different spatial pattern. It has a different covariance structure. So it gets selected out in this analysis. Here you see the cross-frequency coupling modulation, which perfectly reflects the actual peak frequency. And here you see the cross-frequency coupling at electrode POZ, which totally missed it. It didn't even, maybe this is where it should be here. It should be somewhere around here, but that's even a statistical Z-score of around 0. And in fact, this measure is incorrectly identifying this higher frequency gamma as being cross-frequency coupling, which is an alpha error. Basically, it's just being driven by the higher power, plus the non-stationarities that I included into this simulation. Finally, here you see the theta component in blue and the gamma component power time series. So this is the filtered theta signal here in blue, and in the orange color shows the higher frequency gamma power time series. So they match quite well, and it's not surprising. That's how I simulated it. However, when you look at the same analysis with the channel data, you see that, it just doesn't look so nice. The problem here is that at the channel level, all of these signals plus the noise are all getting mixed together. So the individual channel data is not really able to separate these different sources. So in the paper, I have more simulations that you can look through for different ways of setting up the analysis based on slightly different assumptions about the data. But I just want to skip to showing two quick proof of principle examples in empirical data. Of course, in empirical data, we don't really know what the ground truth is, but we can evaluate relative to some null hypothesis testing. So what I did here was take some data from an epilepsy recording. So these are, this was a patient who had 10 electrodes implanted into the medial temporal lobe, which includes the amygdala, the hippocampus, mostly in the hippocampus, and possibly a little bit in the surrounding cortex. And then I took, from that same patient, the electrode on the scalp, CZs, that's at the top of the head, and looked at cross-frequency coupling between the medial temporal lobe, all 10 electrodes, and a CZ, and we take CZ as a measure of the kind of frontal cortical activity in the theta band. So I looked at theta in CZ relative to all the frequencies in medial temporal lobe. So here you see the results of this multivariate cross-frequency coupling measure. It looks like there's some nice peaks here around 65-ish hertz. Maybe some other cross-frequency coupling here, although that's a little bit close to the line noise frequency. And here what you see in the dotted lines is the statistical threshold based on null hypothesis testing. So of course we don't know what the ground truth is, but this is statistically significant. Here you see a time frequency plot of the component, the medial temporal lobe component data relative to the theta peak in CZ. So here you see this modulation, and this is the time course of this frequency around 65 hertz. This is the component weighting, so this shows you where in the hippocampus or where in the medial temporal lobe is driving these effects. So this is one example. I want to show another proof of principle empirical example. This is actually from spike field coherence. So you can also adapt this method to look at spike field coherence, which in my opinion is basically just a different way of thinking about cross-frequency coupling. It's sort of a sister analysis to cross-frequency coupling. So these are data from the Bujaki lab, which I got from CRCNS. The details of the data set are in this paper here. And essentially we have spikes in the hippocampus, and we have LFP in the hippocampus and in the medial prefrontal cortex. So you can align all of the data. These are the hippocampal channels here. These are the PFC channels here. You can pick a neuron or a unit and align the LFP data, and that gives you this spike-triggered average. So what I did was take this a bit further, take a multivariate approach, and in this case I expanded it to look for not only spatial patterns in the signal, but spatial temporal patterns. And this was done by something called delay embedding of a covariance matrix. It ends up giving really large covariance matrices where the space and time are kind of all embedded into a covariance matrix. So, and actually what was interesting is the largest component looked like this, which is pretty clearly an artifact. So there's this spike field artifact that's introduced by the large amplitude of the action potential on the LFP signal. And that artifact actually gets isolated into a specific component. So you can ignore this component, or if you want, you could reconstruct the data without this component to remove that spike field artifact from the data. So this is what the spatial temporal filters look like. I don't have enough time to talk in a lot of detail about how exactly to interpret these, but I do want to highlight that these spike-triggered components are revealing patterns in the data that are not necessarily easily visually apparent just in the spike-triggered average. Now, to be clear, the spike-triggered average is a valid analysis method. These data aren't wrong, but underlying these data, these data, this spike-triggered average, is actually pooling over a lot of distinct spatial temporal patterns that are really there in the data, but are just averaged over, and so they're kind of invisible in the spike-triggered average. So there's a lot of other dynamics that can be recovered from these data by using this multivariate analysis approach. So to summarize, cross-frequency coupling is a really, really interesting phenomenon. I and many other people are excited about cross-frequency coupling findings and methods, and I think cross-frequency coupling certainly deserves more empirical investigations and theoretical investigations. However, that said, I think we need to reconsider the dominant approach of univariate cross-frequency coupling measures, which, as I mentioned, are not necessarily flawed. They're not intrinsically bad, but they actually measure I think they reflect a mixture of true cross-frequency coupling and non-sinusoidal waveform shapes. So I argue that we should start moving from these univariate approaches to multivariate approaches, where we can leverage spatial patterns in the signal and basically just ignore these non-sinusoidal waveform shapes. They don't have to matter for the multivariate approaches. So I introduced one particular approach based on generalized eigendecomposition. There are other multivariate cross-frequency coupling methods out there in the literature. And again, I don't want to argue that this approach that I like is the best one or the only one, but it's the one that I'm focusing on here. But a more general point is I believe that the methods that are most promising, the methods that should give us the most biological and neural insight, the methods that are most meaningful, are the ones that have to start from theory and physiology, not the methods that start from math or signal processing perspectives. And the final thing I want to say is that if you look up the main paper that I've been citing throughout the second half of my talk, this includes much more discussions, of course, of the method and the ways to use it, but also a lot of code and sample data, so you can explore the methods, including all the simulations that I showed yourself, and I definitely encourage you to do that.